Once again, we are here in the translation process. So we all know that from the nucleus DNA replication happens, and then after we have the DNA replication followed by the transcription by the mRNA. So after there is already a transcription of mRNA, the single-stranded, like this, a single-stranded mRNA will then, together with the 5' cap and the poly A tail, they will go out from the nucleus and then they will go to the ribosomes. And the ribosomes now is the site where protein synthesis happens, or amino acids. Okay, so they will go directly to the ribosome. So in the ribosomes you'll find there the tRNA, transfer RNA, and the ribosomal RNA. So the ribosomal RNA, this is the ribosomes capable of synthesizing proteins. If we have here the single-stranded mRNA, okay, I'm gonna have this one, single-stranded mRNA. The ribosomes, the rRNA will help in decoding the sequence of amino acids. For example, usually the start codon starts with AUG for mRNA, and that is methionine. Always find this codon during the start AUG. So the start codon will start, for example, AUG, and then followed by, I'm sorry, I'm going to erase this one. Start. So let's start with, for example, the mRNA is UAAC followed by UUA. Okay. So automatically the ribosome or RNA will pair up the nucleotides. So the tRNA is just all around here, and they have different kinds of codes or nucleotides and nitrogenous pairs. Sample, this one pairs for AUG and this one pairs for AAU. So the transfer mRNA looks like this. This one. So this is the transfer mRNA. This is the tRNA and this is the code like this, and this is now the amino acids attached after they are being expressed. So the ribosomal units binds to mRNA and code for methionine located in the anticodon. So this is now I will use this one. AUG. It starts with here because this is a start codon. So the tRNA will pair up with this, and the mRNA will now have a transcription. It will be now transcribed. So as what I've said, AUG is an anticodon, UAC for tRNA. So the start code of mRNA is AUG, which is an amino acid for methionine. So during the translation process, larger ribosomal subunit binds to the smaller unit S1, forming a ribosomal complex. The tRNA binds to the first active site. This one is the first active site. Active site. And the second one, translation now will begin. So this is the second active site, binding site, and then this is the catalysis site. The catalytic site, this is the site where catalyzes or hydrolyzes methionine, and the next, if there is another, for example, this is leucine, if there is another protein here, the catalytic sites will bind both so that they will form a polypeptide chain or a series of amino acids connected to one another, and that is because of the catalytic site. While the active site, the first active site will bind, the second active site will pair later, and this is the large ribosomal subunits, the big circle. So during the translation process, the second codon of mRNA, like this one, the second codon of mRNA matches the anticodon, this is the anticodon, and then the second, and then the catalytic site will then bind the first amino acid to the second amino acid, and then after this will be bound through the catalytic site, this area portion here will be released back to the system. So this is how it happens. It catalyzes and it forms a peptic bond or a peptide band. So the two amino acids using dehydration synthesis. Remember this one. Dehydration, when you say dehydration, is taking away of water to the bonds, or hydrogen. It's taking away of hydrogen to the bonds. Remember, they are bonded by hydrogen bonds. So take away hydrogen so that it will form a peptic band between them, and then it will connect. Then the process continues, so on and so forth. So the first tRNA now, this one, who came here from methionine, goes to find another methionine in the cytoplasm. He will be searching again for another methionine in the cytoplasm and binds there to the methionine example in the methionine, and then go back again to the system later for the translation process. So the mRNA chain shifts over one codon, placing the second codon CA over the second, and then it moves one codon to the right. A tRNA with an anticodon GUA matching the exposed codon CAU, this one, GUA, it matches with the anticodon CAU, moves into the ribosomes. So this one, GUA, is known as histidine, so it will go against histidine, and the catalytic site will then again hydrolyze and then form peptic band. So the new chain would be now methionine, valine, and histidine. So during the translation process, a new peptide bond forms between valine, histidine, and the catalytic site here. So the tRNA that carried valine will detach. Well, this is where valine is, together with methionine. They were catalyzed, so this is now where they went. They were connected through peptide bond because of the catalytic site. Okay, and then what will happen again to this? This tRNA will now go back to the cytoplasm and look for another valine to attach himself, and then go back again to the ribosomes. So remember, we have the start codon. In mRNA transcription, in mRNA transcription translation, we always start with AUG, the codon AUG, which stands for methionine. While the codon is being translated, methane, valine, histidine, there's a series of codon which is called the stop codon. WAG, and UGA. These are the three stop codons that when they are encountered, the translation process will be ceased. It will stop. So it will message now, oh, stop. So this will be now the final peptide chains of amino acids. And then what will happen to these final amino acids is one, they will go and move down to the ER, endoplasmic reticulum, for further processing. Remember, these chains of amino acids will be undergoing in a process called folding or protein folding. The initial met is removed and the chain is folded into its final shape. So the ER met will be removed and then the remaining will undergo folding. Proteins are being folded in order to make different kinds of structure. So as a summary, DNA complementary strand, it will be undergoing transcription to produce the same strand, and then during the transcription, one of the strand of the DNA, it will be transcribed to make into mRNA, and during this time T will be paired with A, while A will be paired with U. And then after the mRNA, it will go to the ribosomes, where tRNA will pair up the anticodons. So this is now what we call the translation. And after the translation, this is now what we call the protein synthesis, or the synthesizing of amino acids. So as easy as that, is the summary. Now, there are some ways that genes also have fault, and these are genetic errors, or what we call the mutation. So look, why does there is a need for them? Why there is a need to fold the protein? Because it looks like this. The protein folding, it depends on the function. This is the single-stranded amino acid, or the polypeptide chain. Methionine is getting rid of the start codon, and then it will allow it to fold into different structures. One example is the hemoglobin structure, the one that is found in your blood. The structure defines the function of the cell. So in mutation, it only affects few somatic cells and turn into cancerous, and mutation affects a sex cell can be passed on to the offspring. So in the process of meiosis, if there is a defect in the sex cell, then it can be passed through genetics. There are three types. I'm going to explain here three types. The point mutation. The point mutation is a base substitution that may or not code for different amino acid insertion and deletion. Insertion